I'll be talking about antigen. By the end of this session, please don't talk. By the end of this session, you all should know what is antigen and what is heptane. And the factors influencing immunogenicity. But you are talking, I'm just requesting you all not to talk. Don't talk among yourself, it disturbs me. Sit down. So, definition of the antigen and heptane you should know. Then you should know all the factors which are influencing the immunogenicity of the antigen. Then the biological classes of antigens and the super antigens. All this I'll cover in this lecture. Please. Now, antigen is defined as any substance that when introduced in a person will have immunogenicity and antigenicity. These are two terms, both of which are important for antigens. Now, immunogenicity means the ability of an antigen to form antibodies or give an immune response. The immune response can be humoral or cell mediated. So, an antigen will have a reaction with the B cell, which will then become the B cell. What will the reaction be? It will become an effector B cell or the plasma cells. The plasma cells are cells which will make the antibodies, and plus it will also form memory B cells. Then antigens also react with T cells, and the effect is the formation of effector T cells, that is the helper T cells and the cytotoxic T cells, plus it will also form memory T cells. So, antigen is the substance that satisfies immunogenicity more appropriately. They will be known as immunogens. Okay, immunogens can be antigens, but all antigens may not be immunogens. Antigenicity is the immunological reactivity. What I was talking was initially we were talking about immunogenicity. Now we are talking about antigenicity. This is immunological reactivity. When the substance, when the foreign substance which is an antigen causes immune reaction, the antigens will also react. That is, if the antibodies are formed, the antigens will react with the antibody. This is the ability of an antigen to combine specifically. The word specific is important. The antigens will combine with the antibodies which are produced in response to that antigen. They will not combine with antibodies which are produced by other antigens. So, this is a specific reaction, and it will also react with the T cell surface receptors. Okay? Then all molecules having immunogenicity property also show antigenicity, which means that if they produce antigens, they will react with the if they produce antibodies, they will react with the antibodies. But there are certain substances which are antigenic, which means that they will react with the antibodies but not produce it. They are half antigens, they are known as heptanes. They will not initiate the process of producing the antigens, but they can react with the antigen produced by the complete antigen. Now, epitopes are the smallest unit of antigenicity. The antigen itself is a bigger molecule on which there are small areas which may be of 4 to 5 amino acids. You know, all the proteins are made up of amino acids. So, this epitope, which is the reactive part of the antigen, it is made up of 4 to 5 amino acids or monosaccharide residues and is capable of sensitizing the T and B cells. I told you B cells will form the plasma cells and memory cells, and T cells, when react with antigens, they also form T helper cells and some memory cells. So, now specific site of an antibody that reacts with the corresponding epitope of an antigen is called paratope. So, epitope is the region of the antigen and paratope is the region of the antibody which will combine with the epitope. Heptanes are low molecular weight molecules that lack immunogenicity. That means they cannot induce immune response. They cannot form the antibodies, but they retain the reactivity part. That is, they can bind to the specific antibody or T cell receptors. Heptanes can become immunogenic when they combine with large protein molecules called carriers. So, heptane was a half antigen. It can combine with the antibodies but cannot produce antibodies. But if it is combined with a carrier, it can also induce the process of forming antibody and react with it also. There are complex heptanes and there can be simple heptanes which contain only one epitope. You all know what is an epitope? It is a sequence of 4 to 5 amino acids which will form the specific antibodies. Epitope is present on antigen, and where was the paratope? On the antibody. So, simple heptanes contain only one epitope, whereas in complex heptanes they may have two or more epitopes. Now, antigen and host relationship. This is based on antigen, which can be grouped into two groups, self or autoantigens and non-self or foreign antigens. As the name suggests, self or autoantigens are the antigens of the person himself. They may not be immunogenic. The person may not respond to the antigens in the form of forming antibodies because of the immunological tolerance. Now, hosts do not react to their own antigens because of immunological tolerance. There are certain areas where which are not exposed to the immune system during the formation of the embryo, like the inner part of the eye, and all those proteins which are inside will be antigenic. And even the sperms, they are also not exposed to the immune system in the early part of the life, and so immunological tolerance does not develop, and they are self-antigens. Sometimes the self-antigens are biologically altered. For example, in cancer cells, the antigens may be altered. The amino acid sequence may be altered, and hence they may become immunogenic, and antibodies may form against self-antigens. Then there are non-self or foreign antigens. They are of three types based on the phylogenetic distance to the host. Alloantigens are, do you know what is alloimmunity? The alloantigens are the antigens present on the cells of the same person, and these are important for histocompatibility. When there is an organ transplant like HLA typing, MCH, etc., in this case alloantigens are important. Some antigens may be present in some persons and may cause reaction in the other person. Then isoantigens, these are antigens like blood groups. Every person will have one blood group which may match with some people of the same species. In this, the same species will have any one of the antigen, like A blood group or B blood group. What is O blood group? Do they have any antigens? So then there are heteroantigens, which are present in two different species. Here the same antigen, same amino acid sequence is present in two different species, and the antibodies formed by one species will react with the antigen of the other species. These are known as heterophyll antigens. The antibody produced against antigen of one species can react with the other and vice versa. Now, there is a diagnostic application of heterophyll antibodies. The examples are Weil-Felix reaction in typhus fever, where the antigen produced by the rickettsia, which produces typhus fever, antibodies produced by rickettsia will react with the Proteus species, the antigens of Proteus species. So rickettsia you cannot grow. You can have the antigen from Proteus species, react it with the serum of a person who may be suffering with typhus fever. If it reacts, the test will be positive. This is known as Weil-Felix reaction. Then the other tests are Paul-Bunel test for infectious mononucleosis, and still other test is cold agglutination test for primary atypical pneumonia. Now, factors influencing immunogenicity are the size of the antigen, the chemical nature of the antigen, susceptibility of the antigen to the tissue enzymes, structural complexity. foreignness to the host, and genetic factors. All these factors, they influence the immunogenicity of the antigen. How immunogenic is it? Now, optimal dose of the antigen is essential for it to act as an antigen and form the antibodies. Route of antigen administration is also important. How many doses of antigen is received by the host is important. Multiple antigens, if given together, is important, and effect of prior administration of the antibody in the to the person is also important. Now, larger the size, that is, if it is more than 10,000 Dalton molecular weight, for example the hemoglobin, they are more potent as immunogens. They will form more antibodies or will have more immune response. Then the chemical nature of the antigen is also important. Proteins are stronger immunogens than carbohydrates, followed by lipids and nucleic acids. So, first is the protein, then lipids, nucleic acids. Carbohydrates are also immunogenic but less immunogenic. They will form less immune reaction. Now, susceptibility of the antigens to the tissue enzymes. Only substances that are susceptible to the action of tissue enzymes are immunogenic, because the enzymes will degrade and then more epitopes will be exposed. Now, degradation of the antigen by tissue enzymes produces several immunogenic fragments having more number of epitopes which are exposed for the formation of antibodies. Then the structural complexity. Simple homopolymers made up of single amino acids lack immunogenicity. Polymers made up of two or more amino acids are immunogenic. They are more if the structure is complex, it is more immunogenic. Then foreignness to the host. This is the key factor which determines immunogenicity. Higher is the phylogenetic distance between the antigen and the host, more is the immunogenicity. So, within a species, there will be less immunogenicity. If the tissue is from other species, then it will be more immunogenic. Distance in the phylogenetic tree is also important. There were some vaccines which were produced in duck egg. So, those vaccines were more immunogenic. They would cause more reaction than the actual help as a vaccine. So, if the antigens are of different species, they will be more immunogenic. The genetic factor. Different individuals of a given species show different types of immune response towards the same antigen. Some may be responders, some may be slow responders, and some may not respond to the antigen. You all can understand this, that some people are allergic to some pollens, others are not, and some develop hypersensitivity to these pollens or other proteins and others do not. So, this is because of the genetic makeup of the person. Then optimal dose of the antigen is important. If the antigen is given in very small, for different antigens, it is different. Some antigens need a larger dose and some can react in a smaller dose also. Route of antigen administration is important. Generally, parenteral route is important and is more immunogenic depending on the type of the antibody which is produced. If it is an IgA type of response, then it will be more in the oral intake, and you all know any vaccine, oral vaccine? There is one oral polio vaccine. How are the most of the vaccines given otherwise? By parenteral route. Most of them are as injections because it gives more immunogenicity. Repeated dose of antigens are important. If they are spread over a period of time, it will generate an adequate immune response. You all know in COVID you must have heard the first dose and the second dose is given after 6 to 8 months, maybe in this thing, influenza. How long does it, what is the speciation of the vaccine in influenza? लगवाते नहीं है क्या वैक्सीन? चलो, anyway, this is due to the role of the memory cells in the secondary immune response. So memory cells should develop so that the secondary immune response can be had by the recipient of the vaccine. Now, if there are multiple antigens administered simultaneously, the effects may vary. Antibody response to one or the other antigen may be equal or diminished due to antigenic competition or enhanced due to adjuvant-like action. Can you tell me a multi-disease vaccine? DPT. So they enhance they act as adjuvants, and the other is MMR. So there are some vaccines which can be given together, they act as adjuvants, and there are some vaccines which cannot be given together because of the antigenic competition. Primary immune response is more susceptible to get suppressed than the secondary immune response. So generally after the first dose of the vaccine, a person may get the disease, but after 2 or 3, whatever number of doses are recommended, as you have seen in COVID, what was the recommendation first initially? Of 2 doses. Now the booster doses are being given so that the immune response is not suppressed. Any substance that enhances the immunogenicity of an antigen is known as adjuvant. This is added to the vaccines to increase the immunogenicity of the vaccine antigen. The examples are like aluminium hydroxide or phosphate, then mineral oils like liquid paraffin, then Fruent's incomplete adjuvant or a Fruent's complete adjuvant, which is a mixture of Fruent's incomplete adjuvant plus suspension of killed tubercle bacilli in oil phase. Then examples of adjuvants are lipopolysaccharide fraction of the gram-negative bacilli. Then for Mycobacterium bovis, what is Mycobacterium bovis? Yes, it's a bacteria. It is the tubercle bacilli for bovis stands for cattle. No, not for human beings. It is not Mycobacterium tuberculosis. It is Mycobacterium bovis. Then toxoids, diphtheria toxoid and tetanus toxoid. They act as adjuvants of Haemophilus influenza B vaccine. Then non-bacterial products also act as adjuvants. These are silica particles, beryllium sulfate, squenil and thimerosal. How do the adjuvants act? By delaying the release of antigen. All the antigen is not released in once. So it is slow release of the antigen then they also activate phagocytosis by activating the T helper cells and by granuloma formation. How will the granuloma formation help? You understand what is granuloma? Do you understand what is granuloma? No? What is the inflammatory response? Do you all know? Have you read inflammation? Now, depending on the mechanism of inducing antibody formation, antigens are classified as T cell dependent antigens and T cell independent antigens, TD and TI antigens. Most of the normal antigens are T cell dependent. They are processed and presented by the antigen presenting cells to the T cells, which leads to T cell activation. So the antigen, when it enters the body, it is phagocytosed. It is a foreign substance, so it will be phagocytosed by the phagocytic cells. They digest the antigen and then present the antigen on the surface of the phagocytes, that is antigen presenting cell. So it will present the epitopes on the cell surface, which will then react with the T cells and leads to T cell activation. The activated T cells, they secrete cytokines that in turn stimulate the B cells to produce the antibodies. The output is the production of antibodies. So, T independent antigens are such as bacterial capsules, flagella and lipopolysaccharides that do not need the help of T cells and antigen presenting cells. They directly bind to the immunoglobulin receptors present on B cells and stimulate the B cells polyclonally. The antibodies are produced by which cells? B cells. So, in T cell dependent antigens, they were presented on the antigen presenting cells, which was then recognized by the T cells. T cells produce cytokines which induced the antibody production by the B cells. In T cell independent antigens, they do not undergo all this process. They directly go and bind to the immunoglobulin receptor on the B cells, and the B cells are stimulated to form the antibodies. This leads to increased secretion of non-specific antibodies, which are called hypergammaglobulinemia. What are antibodies? The antibodies are immunoglobulins. If they are produced more, the condition is known as hypergammaglobulinemia, and this is a non-specific condition where the antibodies may not be very binding specifically with the antigen. Now there is one thing known as superantigens. The unique feature of superantigens is that they can activate T cells directly without being processed by the APCs. Do you remember how the T cells were activated initially? The antigens were engulfed by the antigen presenting cells. Then the antigens is a variety of biological class of antigens which do not get processed by the APCs. The variable beta region of the T cell receptor appears to be the receptor for superantigens. There is a beta receptor on the T cells which the antigen will bind directly with this beta receptor and act as the superantigen. Now, non-specific activation of T cells because of this combining of the antigen with the beta receptor, it leads to release of cytokines which can activate B cells polyclonally. Polyclonal would mean that many type of B cells, not the specific ones. All type of B cells will be activated and there will be secretion of non-specific antibodies. The examples are toxic shock syndrome toxin, which is produced by Staphylococcus. This is a staphylococcal toxin. Then there is some streptococcal toxin also. It is also, superantigens are also present in Yersinia, Enterocolitica and pseudotuberculosis. There are viral superantigens also present in Epstein-Barr virus, cytomegalo, rabies, HIV, and there are fungal superantigens also in Malassezia furfur. Malassezia furfur is an important fungal infection in HIV. Now, the diseases associated with superantigens are toxic shock syndrome produced by Staphylococcus and food poisoning, also scalded skin syndrome, in which the skin appears to be burnt because of this toxin. So, it is called as scalded skin syndrome. Then there are rare conditions known as atopic dermatitis, Kawasaki syndrome, psoriasis, and acute disseminated encephalomyelitis. So, it can lead to encephalomyelitis also. It becomes very serious. So with this we'll finish